Good morning, everyone. Last class I have discussed about multiple allelism. Last class I have discussed about multiple allelism and codominance, difference between codominance and incomplete dominance, and chromosomal theory of inheritance. So chromosomal theory of inheritance was proposed by chromosomal theory of inheritance. Chromosomal theory of inheritance was proposed by Sutton and Boveri. Sutton and Boveri proposed the chromosomal theory of inheritance and compare Sutton and Boveri compare the Mendelian factors. Compare Mendelian factors with behavior of chromosomes in meiosis and meiosis. Here chromosomal theory of inheritance was proposed by Sutton and Boveri to compare Mendelian factors with behavior of chromosomes in mitosis and meiosis. So according to law of independent assortment, the chromosomes get segregate and assort independently during meiosis or during gamete formation. Sutton and Boveri, both of them observed independently the similarities between in behavior of chromosomes and Mendelian factors. Observe the similarities between behavior of chromosomes and Mendelian factors and proposed two concepts. So first one is the homologous pair of chromosomes, and allelic pair of Mendelian factors. So homologous pair of genes and allelic pair of Mendelian factors. Or segregate and pass into different gametes It's one. So homologous pair of genes and allelic pair of Mendelian factors. Assort or segregate and pass into different gametes. Okay. Then second point is, see, the pair of genes and allelic pair of Mendelian factors segregate and pass into different gametes. Second one is homologous. Homologous pair of genes. Homologous pair of genes and homologous pair of genes and allelic pair of Mendelian factors. Mendelian factors observed independently. During meiosis. So this is the best example, correct, for the chromosomal theory of inheritance. See, homologous pair of genes and allelic pair of Mendelian factors segregate and pass into different gametes. And homologous pair of genes and allelic pair of Mendelian factors assert independently during meiosis. Clear? Next, so chromosomal theory of inheritance. Best example is dihybrid cross. Cross in pea plants. So in dihybrid cross, Mendel crossed two pair of contrasts. So Mendel considered in dihybrid cross, two pair of contrasting characters. These two pair of contrasting characters are controlled by two pair of alleles. Two pair of contrasting characters are controlled by two pairs of alleles. These two pair of alleles are present on different chromosomes. These two pairs of alleles are present on two different chromosomes. And these two chromosomes, these chromosomes are assemblage in metaphase first of meiosis. These chromosomes are assemblage. Assemblage means aggregate at metaphase first of meiosis. And after assemblage, these two chromosomes get separated into possible ways, and as well as the genes present in them in four possible ways. As a result of this, there are four types of gametes. As a result of this, four types of gametes are formed in equal proportion. That's why in F2 generation, along with parental combination, two recombinant type are also produced in F2 generation. So in F2 generation, along with parental type, we know that new combinations are also appeared. So yellow round and green round. Yellow wrinkled and green round. These are the two combinations. The recombinant type appeared in the F2 generation. Clear? Last class I will discuss. So linkage. What is linkage? Next, linkage. And recombination. Linkage and recombination. Linkage and recombination. See, so different genes can control different characters. The genes may control different characters. The such genes are present in may either same chromosome or different chromosomes. Genes for different characters. Genes for different characters. Genes for different characters may either present on same chromosome or different chromosomes or different chromosomes. So genes for different characters. Genes, we know that the different characters are controlled by different genes. So either present on same chromosome or different chromosome. See, according to the law of independent assortment, when the genes are present on the different chromosomes, according to the law of independent assortment, when the genes are present on different chromosomes, assort independently. The genes present on different chromosomes assort independently. On the other hand, if the genes are located very closely, on the same chromosome, they are tend to inherit together. So then, linkage. What is linkage? So linkage is the phenomenon of inheritance. Linkage is the phenomenon of inheritance of inheritance of linkage is the phenomenon of inheritance of two or more genes together. Two or more genes are together present in the same chromosome. Present in the same chromosome. From one generation to another generation. Is called linkage. The phenomenon of inheritance of two or more genes together present in the same frame. So inheritance, the two or more genes are inherit together. They are passed. Inherit means passed together. They are present on the same chromosome from one generation to another generation is called linkage. So linkage was discovered by T.H. Morgan. Very first, the linkage was discovered by T.H. Morgan in Drosophila melanogaster, fruit fly. Drosophila melanogaster. And the genes, the linked genes are get separated. The separation of linked genes is due to the breakage of chromosomes and reciprocal exchange of broken chromosomal segments. between homologous chromosomes is called linkage. Linkage. Such genes are called linked genes. Such genes are called linked genes. So the segregation of is called linkage. Such genes are called here linked genes. Such genes are called linked genes. Then separation of linked genes is due to breakage of chromosomes and reciprocal exchange of broken chromosomal segments between homologous pair is called crossing over. It results in the genetic recombination. So linkage are of two types. Linkage are of two types. Complete linkage, and incomplete linkage. Complete and incomplete linkage. Complete and incomplete linkage. So first I will discuss. So best example linkage is dihybrid crossed in Drosophila. So both for complete linkage and incomplete linkage. So examples of dihybrid crossing Drosophila. Dihybrid crossing Drosophila. For chromosomal theory of inheritance, best example is dihybrid crossing pea plants. But linkage for complete and incomplete linkage, best example is dihybrid crossing Drosophila. So first one, complete linkage. First one, complete linkage. First I will discuss about complete linkage. So complete linkage. Complete linkage. Example, dihybrid cross in Drosophila. So what is complete linkage? What is complete linkage? What is complete linkage? When the genes are placed very closely. When the genes are present very closely. When the genes are present very closely. When the genes are present very closely, shows low recombination. Low recombination and produce low recombination and produce more percentage of parental progeny. And less percentage of recombinant progeny. See, when the genes are present closely, shows low recombination. So, low recombination and produce more percentage of parental progeny and less percentage of recombinant progeny. So, definition for complete linkage. Next, you take the cross. Take the cross. Mendel crossed brown body, red-eyed male, brown body and red-eyed male, crossed with white, yellow body and so white-eyed female, This is parental tag. This is the parental tag. So brown body. So T.H. Morgan crossed the brown-eyed and red-eyed male with yellow body and white-eyed female. So brown body. Clear? Morgan crossed brown body and red-eyed male with yellow body and white-eyed female. This is the phenotype, parental type. So brown, body color brown. Brown is dominant over yellow. Brown is dominant, yellow is recessive, and red is dominant and white is recessive. Clear? Then genotypes. So male dissimilar gametes X and Y. Female similar gametes XX. This is genotypes. Next, gametes. Here, gametes similar XX. Gametes. This is F1 generation. F1 generation. See, brown body, the offsprings of brown body and red-eyed females. Brown body, red-eyed females. Here, yellow body and white-eyed males. Then the F1 generations. When the F1 generation intercrossed. When the F1 generation is intercrossed. When the F1 generation is intercrossed. In F2 generation, we obtained, in F2 generation, we obtained 98.7% of parental time, and 1.3% of recombinant. So then, when F2 individuals are intercrossed, F1 individuals are intercrossed. In F2 generation, we obtain 98.7% of parental type and 1.3% of recombinant type. This is because the genes are closely associated or closely located on the same chromosome. That's why it produces high percentage or more percentage of parental type and less percentage of recombinant type. This is the best example for complete linkage. Next, incomplete linkage. Incomplete linkage. Right now, second one is incomplete linkage. Example, dihybrid crossing Drosophila. So what is incomplete genes? When the genes are located, when the genes are placed far away, means loosely. When the genes are placed far away or loosely, it shows higher recombination type. And it produces more percentage of recombinant type and less percentage of parental type. This is called incomplete linkage. When the genes are placed far away, means loosely, on the same chromosome, it produces higher recombination time. It means more percentage of recombinant type and less percentage of parental type. Take down the cross. So next, to take down the cross. Model crossed red eye, normal wing and red eye and normal wing male. TH Morgan crossed red eye and normal wing male into white eye crossed white eye and miniature wing of female. This is phenotype. White type, miniature winged female. So genotypes. Next, genotypes. Genotypes. So male dissimilar gametes. And similar female similar gametes. Genotypes. Then gametes. Then gametes. F1 generation. This is F1 generation. F1 generation. So F1 generation is intercrossed. F1 generation is intercrossed. F1 generation is intercrossed. In F2 generation, we obtain F1 intercrossed. Then in F2 generation, we obtain 62.8% recombinant time. 62.8% recombinant time. And 37.2% parental time. See, 62.8% of progenies so in F1 generation to cross F2 generation, so 62.8%, so parental time, and it is recombinant time. 37.2 is recombinant type. So these two in F1 is interposed. F1 interposed. In F2 generation, we obtain 62.8% parental time and 37.2% recombinant time. Okay. So it completes. So linkage and recombination. In linkage, two types: complete linkage and incomplete linkage. So T.H. Morgan, so give example, and T.H. Morgan explains the linkage with example. So Drosophila, so melanogaster.